It is my greatest pleasure to introduce Kaiyi today, who will be talking about his recent paper, EVOLVEpro. Kaiyi is a PhD student at MIT in their biological engineering department. He is triple co-advised by Omar, Jonathan, and Michael, and his research is broadly focusing on discovery and engineering of reprogrammable biological systems to engineer cells. So Kaiyi, we're so excited to have you today, and we are ready to hear more about EVOLVEpro whenever you're ready. Thank you so much.
Right, thank you for the introduction. Let me share my slides. Okay, can you see this? No problem?
Looks good.
Okay, cool. All right. Thank you again. So I'm excited to be here to share a little bit about what we have done using a large protein language model to evolve proteins. As a background, I'm actually not a computer scientist by training. I'm actually a biological engineer and biochemist by training. So I spend a lot of time just discovering reprogrammable system by looking at what evolution has already done. And for the most part, I actually have been benefited by the advancement in machine learning models. And for the most part, I'm a user of these really cool models. An inspiration for doing this work, EVOLVEpro, has been to sort of give back to the community and really thinking about this problem from a biological engineering perspective. What should be the right benchmark and such. So I would say the story really started back in 2021, and a team of scientists at back then Facebook, led by Alex Reeves, actually detailed the development of a foundational model that is trained on protein. So they sort of utilized what has been done and described in the natural language processing field in computer science and just apply this autoregressive transformer-based model to training on protein sequences. And at the time, they described this model called ESM1B, and all they did was actually take all the protein sequences in UniProt and this data set called UniRef, and they do this autoregressively. So they mask a portion of the protein sequences and ask the model to iteratively fill it back. And by doing so, eventually they figured out that the model, just like what they see in the natural language processing field, developed some intuition and capability about the basic biophysics and biochemistry behind protein and amino acids. And they have done some really nice characterization about the embedding space, about how this model is able to encode protein amino acid sequences into a really high dimensional information-rich latent space. And I think as a biological engineer, the coolest part is actually they deployed this model in a zero-shot fashion to rank fitness of protein. Now, if you look at this diagram on the right, what they did in the very initial phase of this model is they just took a deep mutational scanning data set, which biochemists and biophysicists have collected over a range of proteins. So they do this by basically cloning point mutations onto each part of amino acid on a protein and assay their activity in a variety of application settings. And what the team decided to do is they asked whether probability of a certain protein sequence existing in this ESM large language model is actually correlated to the fitness, the actual activity of that protein in a particular assay. And when they do this, they actually compare to a bunch of other models, and they showed that ESM1B or this large protein language model in general do develop some intuition about ranking the fitness or the activity of such proteins. And it has been really exciting, and we fast forward to 2 years later, we have better and better pre-trained large language models, and now this is ESM2, also developed by a team led by Alex Reeves. And people are really amazed by the capability of such large protein language models. And one particular way that people have been using this sort of model is to really, again, take advantage of the fact that you can calculate the negative log likelihood of certain amino acid and protein sequences existing in the model. And by doing so, because the model has seen all the possible protein sequences arise from evolution, then there's this idea that if a sequence is more plausible by the model, then it is probably more evolutionary fit. So in other words, it's a closer to the evolution distribution that exists in nature. And what Brian He currently at Stanford has done is he sort of took this model and then computed the negative log likelihood of different antibody variants. And by doing so, the hypothesis is that the sequences or the variants that has a higher probability is probably more fit. In other words, they might be more stable, they might fold better. And for antibody, the particular objective is, of course, to bind a certain antigen. And when he did this, he actually surveyed a bunch of model-ranked variants for different antibodies, and he showed that if you ask the model to pre-rank this for you, then the probability of success is much, much higher. And if you look at the diagram on the right, he also did this deep mutational scanning database benchmark, where he asked the model to rank the variant effect of different mutations. And by doing so, of course, you can easily beat background sampling. So this has been really cool. And now we fast forward to 2024, which is today. We have even more exciting models that is recently described called ESM3. Now, what ESM3 has done is that it's no longer a sequence-only model. So they start to incorporate other informations and features into the training phase. So in this particular case, they described architecture where they also incorporated structure and function into this pre-training phase. And by doing so, ESM3 actually become generative protein language model. Now, in other words, this model has the ability to basically mask or unmask certain portion of a protein sequences. And by doing so, you can start off to ask the model to de novo design a sequence for you, given a certain backbone or given a portion of protein sequences. And they have done some really nice experimental characterization too. Now, if you look on the right, what they decided to do is they decided to ask ESM3 to de novo design a GFP sequence. Now, the ability to de novo design a sequence that is very lowly homologous to what exists in nature, but still do the same function is very, very useful and an impressive capability. And they show that ESM3 does have this capability. Now, when they ask the model to basically unmask the amino acid around the GFP core, they are able to design different GFP sequences. And obviously, not all of them work. And the top, 1 or 2 seed sequences, they decided to do further experimental benchmark. And now you arrive at this result on the lower right corner of the slide. And what we see is that so this ESM3 GFP sequence is very lowly homologous to the natural one, which is the AV GFP all the way on the left side of the curve. And they are still doing the same biological function. In other words, you can sort of, you can measure the fluorescence intensity. But the problem with this de novo design seed sequence is that it has a really slower maturation kinetic. So now if you compare to AVGFP, you don't see much difference in the fluorescence intensity between day 0, day 2, or day 7. But for ESM3 GFP, you actually see a really slower kinetic. And that's just one problem. The other problem is that it seems like even at day 7, the mean fluorescence intensity you get from this particular sequence is still much, much lower than AVGFP. Now, if we take a step back, evaluate this result from a bioengineering and biologist perspective, actually, we know that a lot of rational engineering have been poured onto AVGFP. And in fact, in laboratories, people don't use AVGFP anymore. Nowadays in the lab, we use EGFP or M neon green or M state gold, which are basically engineered version of this GFP protein, which are much, much brighter, orders of magnitude brighter, to suit a variety of biological applications. And ESM3 GFP in this particular case, even compared to the most native AV GFP, is still not bright enough. So we know there's a lot of room to go about improving these de novo design sequences. And I will give you another example of this. And this work is also published in 2024, done by Proofland Bio. Now, what they decided to do is rather than retraining a new large language model, what they did is they took the existing protein language model and they fine-tuned it on a subset of protein sequences. In this particular case, they are very interested in CRISPR-Cas protein. Now, CRISPR-Cas proteins are RNA-guided DNA nucleases that are very important in a variety of applications, including gene therapy and cell therapy. Now, what they decided to do is they curate a data set of CRISPR-Cas. They call CRISPR-Cas Atlas. And that includes around 5 million different CRISPR proteins. And they just fine-tune this pre-trained universal protein language model on this subset. And the assumption is that by doing this fine-tuning, which actually happens in a variety of computer science applications, you can sort of adapt the model to be more suitable for one particular downstream task. And now when they do this, they find that if you couple this with another RNA model that is able to generate RNA sequences for these Cas proteins, then you really have the ability to de novo design Cas proteins. Now, this is very impressive because RNA-guided DNA nucleases are probably one of the hardest and most complex molecular machinery that exists in nature. And then they did a lot of extensive biochemical characterization around this protein they called open CRISPR-1. And what you find is that quite different from ESM3 GFP, which is not as bright as AV GFP, they decided to benchmark against SP-Cas9. Now, this is sort of the golden standards in the genome editing space. And what you see is that although it's not much better than SP-Cas9, but this particular de novo design sequence is actually has on par cleavage efficiency with sort of this golden standard in what we use nowadays in the lab. So we know this is really cool, and these models sort of have this ability to de novo design these sequences that sometimes could be as good as what exists in nature. Now, from a bioengineering perspective, all these de novo design capabilities are really cool. But in this particular two application settings, if you want to design GFP sequences or you want to design Cas9 proteins, it's actually better if you just look at what nature has already provided for you, right? So nature has already done a lot of filtering for different protein sequences, and we can always just steal these protein sequences or system from nature. And we know that these sequences, when you reprogram them to do synthetic functions, are quite good. And what biological engineers really want is actually a model that can further improve the activity based on what exists in nature already, right? So we want so-called gain-of-function mutants that confer some super physiological activity that we can use in synthetic system, right, to do therapeutics or diagnostic. So that is really the motivation behind why we developed EVOLVEpro. And EVOLVEpro is built on this idea that negative log likelihood or probability of certain protein sequences existing in protein language model might not correlate with the experimental activity. Now, if we think about evolution, right, evolution does not act on an individual level. So evolution does not select for individual protein to be maximal activity. And in fact, evolution almost always punishes for individual proteins to be at their maximal activity, because evolution works on a system level. They want to ensure that when a group of proteins, when a group of genes, they function together, they have to ensure an organism's maximal activity. And in fact, sometimes evolution even works on a population level, right? So you do not want to satisfy individuals' maximal fitness, but you want to satisfy population's maximal fitness. So if we sort of treat that assumption in mind, then we'll arrive at the conclusion that if you simply ask the model to do this in a zero-shot fashion, where you use the probability score of these different variants as their actual predicted fitness or activity, then it might not be correct. And what I decided to do is that I will just take the embedding, which we know are really information rich and very high dimensional, and I will couple this with a domain expert top layer. And now this is a simple regression model, right? The regression model's input is actually protein language models embedding generated by these pre-trained large PLM. And you can sort of feed this into a regression model. And on the other end, the output is actually experimentally measured activity of a protein. And you can consider this as really a versatile usage of PLM embedding, because for different proteins, you might have different domain expert top layers, right? So for each different particular biotechnological application, you can sort of train a unique regression model to couple the experimental activity with the input latent space embedding. And by doing so, the cool thing is that you don't have to retrain this large language model anymore, because that's actually very computationally heavy, right? And the top layer regression model, these are really lightweight regression model that can be as simple as a linear ridge regression. And when you deploy this on top, you can actually rapidly train this regression model to connect the latent space embedding generated by PLM and experimentally measured activity. And more interestingly, you don't need to do this in a zero-shot fashion, because we know that for many of the proteins that we want to evolve, we don't have the ability to massively parallelly screen for their activity. And sometimes you only have very limited throughput, right? So in those settings, what you can do is you can sort of use EVOLVEpro in an active learning format. In other words, in each round, you don't need to get all the data to the model, right? So you can decide to only collect 10 or 20 data points. And you can sort of feed this 10 to 20 experimental data points to the top layer regression model and ask it to fit through the whole distribution, right? And the model will rank for you what's going to be the most likely to be higher activity so that you can get an idea of what you need to test for the next round. And when you do this iteratively in an active learning format, eventually, in a few rounds, the model will have the ability to sort of guide you to certain local maxima or even global maxima. So that's the overall idea of EVOLVEpro. Nothing too fancy, really just sort of connecting a top layer regression model to PLM. Now, there are a few parameters that you can tune in this particular model. And if you think about it, there are three major parameters here. So we have the base layer, which is a protein language model we use to encode protein amino acid sequences into the latent space embedding. We also have the top layer regression model, and you can sort of deploy a range of basic linear regression models or even multi-layer perceptron MLPs in the top layer. And lastly, you sort of have this active learning strategy, right? So how greedy do you want to be? How do you balance exploitation versus exploration? So we sort of benchmark these three parameters using public DMS datasets. And if we look on the left, what I show you is average across 12 different DP mutational scanning datasets. And these 12 datasets are curated in the idea that we want to maximize protein applications. So these 12 datasets include antibody binding, GPCR, kinases. So we want to make sure that the model is not doing good on a particular dataset, but rather it's actually very generalizable across different assays. And when you do this, and we can compare different top layer models' performance versus the active learning strategy. And to our surprise, what we found is that the best performing combination actually arises from using a random forest regressor as our top layer model and using top 10 as the active learning strategy. And you might suspect that you don't want to be greedy all the time, right? Because if you always choose the top K to test for next round, then you might be biased into the model leading you to certain local minima, and you get trapped there. But in this particular setting, random forest regressor actually worked really well in this sort of overfitting scheme, right? So in each round, the model is only able to see 10 to 12 data points, and this is very little compared to the whole landscape. But even then, what we find is that if you always stick to top K, so you want to be maximally greedy, and if you use random forest regressor, which in the literature also has been reported to do very well with overfitting schemes, then this is the best combination. It gives the best average performance across a range of datasets. And we also decided to just really test different protein language models out there, right? And what we used in the very beginning is ESM2 15 billion. So this is the largest parameter protein language model published by the Meta team. And when we compare this to a range of other models, what we find is that these pre-trained language model in general, they outperform, one hot encoded or integer encoded, which is very simple representation of the protein. But if you compare the performance across these different protein language model, right, you actually see diminishing return. So if you compare the 15 billion with the 650 million or the 3 billion parameter, you get very marginal boost in the performance in some data sets, whereas in other data sets, they are sort of just equally good. So this also speaks to the fact that just simply increasing the parameters in this large protein language model might not get you better performance in some cases. And how does this look compared to other models, right? So I have been saying that if you simply trust these negative log likelihood scores in a zero-shot setting from these language models, they do not work in some cases. Now, in this particular case, I benchmarked against the paper I showed you before, published by Brian, and they call this efficient evolution. So if you compare the performance of EVOLVEpro with efficient evolution, what you find is that by leveraging this top layer regression model to connect the PLM with the actual experimentally measured activity, then you can really do much, much better, right? And in the active learning format, of course, the more rounds you have, the better and the better the model is able to decipher which part of the landscape leads to better activity mutants. And of course, you don't have to do this in active learning format like the ones shown on the right, right? You can also deploy EVOLVEpro in a zero-shot fashion. So we call this pre-training, where you sort of, if you have the ability to massively parallelly screen, hundreds or thousands of mutants in a particular assay, then you can simply feed this data in round one, right, to the model, and you ask the model to only run for you once, which mutants are likely to be higher activity. And when you do this, of course, you see that increasing the number of the pre-training data always leads to an increase in the performance of the model, right? And at around 1,000 mutants, then we achieve 100% higher activity candidates for most of the data set being tested. So I showed you basically this overall idea of EVOLVEpro being deployed in a variety of settings. And you might wonder, right, for a variety of different applications, what should be the number of the seed sequences that you should test in an active learning format? Now, we decided to vary this number. So the number of the sequences you test actually in wet lab from 10 to 100, right? And depending on your flavor, depending on what this protein is, and depending on the throughput, you can, of course, adjust this parameter. But we adjust this parameter computationally across these 12 data sets. And what we find, similar to our expectation, is that increasing the number will always lead to an increase in performance, right? In the performance of the predicted activity. And as low as 10 mutants per round, if you do this for 10 rounds, will actually lead you to pretty good results, by the end of the round 10. But if you have the ability to increase this number, of course, it's always better to be on the higher side. And in fact, if you increase this number to 100, then for most of the data set, then we see at the end of round 10, you get 100% predicted higher activity candidates by EVOLVEpro. So all the data I've shown you before is computational benchmark. And although they are very cool, they are very good for you to tweak the parameters in this model. What biological engineers really care about is how do you deploy these in real application settings. So I will show you a few examples throughout the slide. And to start with, I will show you how we deployed EVOLVEpro to engineer a miniature CRISPR nucleases. So SP-Cas9 is very good. It's sort of the golden standard in the industry for genome editing purposes. But there's a problem with it, that the size is too big, right? So they don't fit in AAV vectors, and you have a hard time finding a good promoter that is suitable when you want to do in vivo gene therapy. So a lot of biological engineers have focused on sort of finding these miniature CRISPR nucleases in Cas12 families. And in this particular case, the field has sort of arrived at a range of different Cas12F nucleases that we know are only 1/3 of the size of SP-Cas9. So they fit into AAV vector really well. That allows you to couple them with different promoters to do this in vivo gene therapy. And the problem with these Cas12F nucleases are they are not as efficient as SP-Cas9. So when you sort of reprogram them from bacteria into mammalian cell, what you see is that for most of the locus, they no longer edit the genome. So they have really low editing efficiency. And a lot of rational engineering has been poured onto engineering the guide RNA or the protein. And it's really just, there's no generalizable design rule for engineering this protein. And it's a headache to sort of evolve this protein to a certain threshold that you can use it. So I took a Cas12F protein sequences we have in the lab, which we know are very low efficiency. So it's only around 10% editing efficiency in most of the locus in hexcell. And what we decided to do is that we set up an assay where we ask different Cas12F variants to cleave a certain locus in hexcell. And by doing so, you can sort of survey different cleavage efficiency of different mutations. And we feed this data into EVOLVEpro, and we do this in an active learning format for 5 rounds. And shown here on the data on the right, each data point is actually a mutant nominated by EVOLVEpro. And if you look at round 1, which is just random guesses by me onto this protein about which part of the mutation might make it better, starting at round 2, the model really suggested some better activity mutants compared to wild type. So on the y-axis here, this is fold improvement over wild type, which is what exists in nature. And by the end of the round 5, which we only tested 60 mutants in total, we already arrived at a variance that is around 5-fold more active than what exists in nature. And of course, this particular assay is set up using only one editing efficiency locus in hexcell. And we know that that's hardly generalizable because most of the engineering carried onto these or the guide RNA, they are very locus specific, meaning that the improved variant work in that one particular locus, but they don't generalize to other part of the genome. So we wanted to ask whether EVOLVEpro actually understood how to make this protein globally better at editing the genome, rather than sort of just finding a way to hack that one particular locus. And when we do this, we compare this evolved PSA-Cas12f with a range of published Cas12f nucleases. What we find is that across the board, it's much better than both the wild-type PSA-Cas12f, which is what exists in nature, and also other published or engineered variants of small nucleases. So we know that the engineered mutants are not an artifact on one particular locus. And this is really nice to know. And of course, with that being said, we can sort of benchmark against back to SP-Cas9, which is the golden standard, right? And you can ask these different variants to edit the PCSK9 gene in a liver cancer cell line. And what we see is that the engineered variants is really highly effective. So it has cleavage efficiency on par with this golden standard SP-Cas9. Although the size of this evolved PSA-Cas12f is only 1/3 of what is for SP-Cas9. So you cannot package SP-Cas9 in AAV, but you could do so for this evolved PSA-Cas12f. And that's exactly what we did. So after we know that it works really well in liver cancer cell, we package this evolved variant into AAV and we inject it into mice. And the cool thing is that once this AAV enters the liver, the PSA-Cas12f, of course, edits the PCSK9 gene, causing indel formation in that particular gene and knock the gene out. And you can sort of detect the decrease in the PCSK9 protein level in the serum. And when we do this, we see that it actually works really well. So when you inject the AAV, only after 2 weeks after injection, we are already seeing around 50% decrease in the blood serum PCSK9 compared to PBS injections. So we know this is a success for EVOLVEpro as it is able to evolve this particular protein in only less than 60 tries. And we decided to understand a little bit better about how this particular model is able to evolve this protein. In other words, is there any biophysical basis for these particular mutations? And I think this is a really important aspect of this work is that a lot of these black box machine learning models, they do give you better mutants, right? But most of the time, these mutants are in places where rational engineering would not have engineered it. And there's sort of no biophysical intuition or basis behind explaining why did the model choose a particular residue. And in this particular case, we decided to map the top 3 mutations nominated by EVOLVEpro onto the structure of this PSA-Cas12f. And what we see is that off the board, the K454 particular mutation happens on the RAV-C domain. So RAV-C domain is the catalytic domain of the Cas12f nucleases. And typically, when you carry out rational engineering, the RAV-C domain is always an area of focus because making this particular domain better almost always leads to an increase in the performance of the nuclease. And more interestingly, if you look at this I178 mutations, this particular mutations happens on the alpha helices, right? And what the model decided to ask us to mutate is to mutate this I178 to alanine. And for a fact, we know alanine has a higher alpha helix propensity than the I in this particular wild-type residue. So it's sort of natural for us to hypothesize that maybe the model is sort of understanding how to strengthen the interactions of this alpha helix here. And lastly, this K333 residue being nominated by the model, this is where the protein binds the guide RNA, right? So this is another hotspot in which if you had the structure, then rational engineering will tell you to sort of engineer this part of the protein. So we sort of know that the model is able to decipher these key activity sites in the protein rather than just randomly mutagenizing the protein in places where we do not expect at all. And if you look at the attention of EVOLVEpro, right? So we plot here the cumulative time or the frequency that the model visited a particular residue, then you really see that the model sort of gains attention onto certain location of the protein. In this particular case, there are positions like G147 and E151 that are being visited multiple times, right? So in later rounds, the model asks us to mutate this particular residue to multiple different amino acids in the sense that the model is trying to understand which particular mutation makes it better. So we arrive at this plot in the end. That is, I think, a center plot behind the assumption of whether fitness in evolution actually correlates with activity. And if you look at the plot on the right, the x-axis denotes experimentally measured activity. So this is actual fold improvement of a certain protein over the wild type in a realistic assay, right? On the y-axis, we have predicted fitness by the bottom protein language model. So there are different ways to calculate this. In this particular case, I choose mask marginal score, which is a common function people use to sort of compute the probability of certain sequences. And this has been used again and again to sort of rank different variants in a zero-shot format. And when you compare this, at least across the 60 mutants being tested in this particular assay, then what you find is that there's actually very little or even zero correlation behind the predicted fitness by the language model and the actual experimental measured activity, right? And this points back to this original claim that probability of a sequence being closer to the evolutionary distribution simply might not be correlated with their activity, because evolution might not have selected for very high activity when it evolved that particular protein sequence. And this is, of course, the local landscape. And if you were to extrapolate the top layer regression model to the whole landscape of the single mutant, then what you still see is that it seems like the top layer is not agreeing with the bottom layer negative log likelihood scores. And this points to the fact that, at least from a bioengineering perspective, right, we do need more sequence to activity data to reflect how the model is doing and to properly benchmark these models on their ability to evolve proteins and rank nominations for us. So Cas12f is just one application in my paper, and I think we got really lucky because, of course, the wild type is not really efficient, and we are very easy to engineer this protein because rational engineer can also get you there. And we decided to test the model with a harder task, right? So there are proteins that are already highly evolved, highly engineered in the lab. So in this particular case, what we decided to do is we want the model to evolve a prime editor. So prime editor is a fusion protein. So this particular protein actually doesn't exist in nature. It's made in the lab synthetically by fusing a nickase Cas9 with a reverse transcriptase. And this is a really important therapeutic protein in the field of genome editing because it's sort of the first tool that allows you to precisely edit the genome by either inserting or deleting a few base pairs at a random location in the genome. And we know that a lot of rational engineering have been poured onto this prime editor. So we decided to start not from the wild type natural prime editor. We decided to start from prime editor 2, which is already a very evolved variant from David Liu's lab at the Broad Institute. And now, if you look at this evolution trajectory in round 1, which is random guesses by me, then most of the mutants actually destroy the protein, right? They make it much worse. And this is as expected because this particular protein sequence is already very engineered, very evolved. And over the course of the evolution by our model, right, you immediately see that the model, although on a very slow trajectory, but they are able to sort of figure out which part of this already very evolved protein to mutate to make it better. And in this particular case, it took us 7 rounds, right? So 84 total mutants to get us to a final evolved variance that is around 1.7 to 1.8 fold better than wild type. And although this fold change is very low, but we have to keep in mind that the wild type, as we used here, is already prime editor 2. So this is a very evolved and highly efficient enzyme. So 1.7 fold improvement over it is not trivial. And we also tested this evolved variants by our model across a range of different locus by asking the prime editor to insert different sequences into different genomic regions. And what we see in the end is that it's actually better than the wild type across the board. And I think biophysically, more interestingly, what we see is that a variety of the mutations being nominated by the model actually fall into the RNase H domain rather than the reverse transcriptase domain typically used as an active site for this enzyme. And this is quite surprising because almost all the rational engineering effort have focused on engineering the reverse transcriptase domain. And there's very little effort being focused on the RNA stage domain. In fact, this domain has been regarded as an optional domain. So prime editor can delete this domain and still function. Nevertheless, the model deciphered that a variety of different mutations on this particular domain will actually increase the efficiency of the protein. So this is sort of application setting where the model points to non-intuitive places in the protein where rational engineering would not have carried out to make this protein better. And just another example of very hard to engineer protein, this is a BXB1 integrase, yet another very highly active and efficient enzyme that already exists in nature. And you see again, right, in round one, most of the mutations actually kill the protein. But nevertheless, if you ask the EVOLVEpro to evolve this, then the top layer over a few rounds is rapidly able to identify mutations that lead to beneficial activity for this BXB1 integrase. And of course, this is a very important protein in biotechnology because you can sort of combine this with long-range genome editing technologies to insert arbitrarily long cargos into desired location in the genome. And when we do this, of course, across different locus, we also see much higher efficiency compared to the wild type, showing you the efficiency of the model and also this evolved variants of the integrase. And when we zoomed in onto understanding the biophysical of the mutation behind why the model made the BXB1 better, off the spot, what we see is that most of the beneficial mutations nominated by the model actually cluster around the DNA binding domain. And this is, of course, expected because when you have the structure, rational engineer and structural biologist would also point you to these locations, right? So they really make a lot of biophysical sense behind why these mutations make the protein better. And similar to the proteins I've shown you before, right? The model really developed the intuition about which part of the protein to engineer over a few rounds. And you can see the cumulative frequency increases very heavily for a few residues. And typically, the model takes a few tries at mutating these residues to different amino acid in order to find an optimal particular mutation at that location. And more interestingly, in this particular case, right, if we compare the activity, experimentally measured activity, and the predicted negative log likelihood from the model, we actually finally see a situation where it actually agrees with the literature, right? So in the evolution of BXB1 integrase, what we see at least is that the activity is positively correlated with this predicted fitness by the bottom layer protein language model. And this again points to the fact that for different proteins, you might have different scenarios, right? Where evolution might have really placed emphasis on the efficiency of certain protein. And in this particular case, for the BXB1 integrase, that seems to be the case. Whereas for other proteins, for example, for prime editor or for Cas nucleases, it might not have been the case. So having that flexible top layer regression model really help you generalize across the board when you want to improve the activity of certain proteins. And I will end my talk on one last protein, which I think is the most exciting part of the paper, where we, for the first time, we start to explore multi-objective optimization. And this is really important in directed evolution and also in many of the experimental assays where a given protein does not have one single objective that we want to optimize, right? And when we want to optimize these type of proteins, it is typical that they have multiple parameters that we need to tweak. And they might not be correlated with each other. So they might be independent from each other. And it makes it a really hard task for us to set up assays to properly test these protein mutations, because when you have multiple parameters in mind, of course, your assay has to expand, and you lose sort of the ability to massively screen, thousands or hundreds of mutants. And in this particular case, we focused on T7 RNA polymerase. And this is a really important biotechnological protein because this is the workhorse behind generating mRNA, which go on to become vaccines or Cas9 that allows in vivo gene therapy. And when thinking about optimization of the T7 polymerase, there are several parameters that we need to care about. For one, we need to care about how much mRNA it's able to transcribe. And for the other one, we need to care about what is the percent full-length product, or in other words, what is the percent correct product generated by these T7 RNA polymerase. And lastly, we ask, how many mistakes does the protein make, right? So how many double-stranded RNA and toxic fragments does this T7 RNA polymerase make? And all these parameters are really important when determining whether a mutation onto T7 RNA polymerase is beneficial or making it worse. And in this particular case, the flexibility of the top layer regression model allows us to basically couple the embedding to different objectives, right? And we can sort of weight these different parameters using a custom function. And in this particular evolution regime, we just decided to weigh 1/3 of the weight on the function into each parameter, right? So we care about these parameters equally. And when you ask the EVOLVEpro to evolve these proteins, what we see is that in less than 60 total mutants, we arrived at mutations that are up to 20 to 30-fold better than wild type. And of course, if you only look at this graph, this is sort of a single axis, right? So this is average score across the 3 parameters. And I think it's more interesting to look at the plot below, right, where we sort of project different parameters onto these proteins. And what you see is that if we have on the x-axis immunogenicity, so this is how much double-stranded RNA a protein generates, and on the y-axis we have translation, so how much full-length correct product the protein is able to make. Then what we see is that, of course, starting from the wild type, the round 1, which is a guess by me, most of the time actually destroy the protein, right? They either make it more immunogenic, so they generate more mistakes, or they generate less full-length product, right? So you have less translation. But over a few rounds of evolution, you really start to have the ability to design, sequences that are much, much better than those that exist in nature. And in the end, in the multi-mutant round 6, we arrive at this triple mutant, that are up to 100-fold better in one axis, for example, in immunogenicity. So it produces very, very little double-stranded RNA compared to the wild-type T7 RNA polymerase, which is a very important feature of this engineered T7 RNA polymerase. And of course, we can actually purify these proteins and test this across the different templates, right? And in evolution, we only decided to make one particular luciferase mRNA for testing purposes. But when we purify this protein, we find that the processivity and the generalizability of this evolved variants is really across a range of different mRNA sequences, regardless of GC content, regardless of the length. And when we use more mechanistic assays to decipher why this is the case, we decided to probe how much double-stranded RNA is generated by running an ELISA on the produced mRNA. And we see that this evolved T7 polymerase really generates much, much lower double-stranded RNA compared to the wild type. And in fact, this is on par with the golden standard published by the Moderna about minimally immunogenic T7 RNA polymerase mutants. And biophysically, this also makes a lot of sense. If we look at the structure, right, the top rank mutation, E643, happens to be in contact with the DNA region that serves as the template for the polymerase to make RNA, right? And typically, again, this is a hotspot for engineering. But more interestingly, typically the rational engineering would ask you to mutate these negatively charged amino acids to positively charged amino acid, with the assumption that the positively charged amino acid will be attracted to the DNA, so the protein binds to the DNA better. And this is the fundamental assumption behind a lot of the rational engineering being done on nucleases and DNA binding proteins. But through the evolution, what we figured out is that if you mutate this E643 to lysine, for example, which is positively charged, then it actually destroys the protein, so it doesn't make it better. But the model is able to figure out that if you mutate this particular residue E643G to G, which is a glycine, then somehow this particular mutation magically did make the protein much, much better, both in terms of processivity, but also in terms of the amount of the full-length product and minimal double-stranded RNA generation. And just to show you, how good this is, right? We can make GFP RNA and transfect this into the cell. And here I show you that compared to the mRNA being made by the wild-type polymerase, this evolved polymerase really make much, much better mRNA, right? So at the same dose, you generate a lot more fluorescence intensity compared to the wild-type one. And of course, we can also just use this protein to make clinically standard mRNA, right? Where you have 100% modified pseudouridine bases being inserted into the mRNA. And when we do the bioluminescent imaging in mice, we're also able to figure out that it generates much better mRNA quality than the wild-type ones. So with that being said, I think the conclusion of this model and my paper is really to say this one thing, right? If you have to take home one thing, the message is sometime, and in fact for most of the proteins, right, higher fitness in evolution does not equal to higher activity. Because for a variety of biotechnological application, we want to maximize a protein's activity in a certain assay. And typically, this is not rewarded or explored by nature, right? So there's no reason to expect that these large pre-trained language model will give you the ability to sort of just zero-shot optimize variants for you. And in those particular settings, it's very important to set up these top-layer regression model or other set architectures to actually make the model focus on the experimental measured activity so that you can rapidly evolve a protein for an application. And the hope is that one day, right, I come back to this example of ApoE3 versus ApoE4, where a single mutation in cysteine 112, which is typically what we have in normal humans, gets mutated to arginine, and this becomes pathogenic, right? So a single mutation in ApoE3 can actually change the global structure of this ApoE protein. And then, of course, this structural change is accompanied by a range of activity change for this particular mutants that make it really pathogenic. And the hope is that one day we will have a lot of sequence to activity data to train our models, and the model should develop these biophysical understanding about sort of these minute changes in the protein sequences that leads to large change in their activity. And lastly, I want to acknowledge all the help from the Abdaya Gutenberg lab, and also this is a collaboration with the Weizmann lab at the Whitehead Institute, Hiroshi's lab at University of Tokyo, and also Daryl Irvin and Michael Berman's lab at MIT's Koch Institute. So yeah, with that, I will conclude my talk and happy to take any questions.
Thank you so much. We do have quite a few questions in the chat, so I'm going to go ahead and start reading some of those, but I'm also going to give people the opportunity to unmute if they would like to ask questions too. So the first question that did come up in the chat was, why would you say that ESM3 de novo designs are lower activity? And you guys can also unmute if you want to expand on that question as well.
Right. So that's a really good question. So ESM3 sort of is really impressive at de novo design, right? And I think it's really useful in the cases where you don't know where to start, right? So evolution doesn't point you to a particular sequence for you to start optimizing. Then these de novo generative PLM really shines because they sort of give you a really nice starting point for you to play with. But like I said, right, the original pre-training task for these large protein language models is to sort of look at the distribution that already exists in nature, right? And by doing so, all they learn is sort of what happens during evolution. And there's no reason to believe that you can actually learn, right, the activity for a certain protein because it might be sort of underrepresented or because evolution just doesn't reward for, for example, in the case of GFP, maximal bright intensity. So that might be the case why these generative models, de novo design sequences are not as good.
Awesome. The next question I have is from Justin. He asked, is it possible that some of this performance difference across PLMs is driven by the difference in embedding dimensions?
Yes, I do think so, and this is a very good question, right? So during the early development phase of our work, we explored a range of ways to utilize the embedding from ESM2, for example. So what ESM2 does is, of course, it generates a L times 5200 matrix, right? So for each amino acid, it generates a vector. And there are different ways to use this, right? Some people use the raw embedding, some people pull it by averaging across all the amino acid. Some people take the max, some people only consider the position being mutated and use that particular one vector. And when we compare this, right, either by using PCA to shrink them down or just using the raw embedding, we didn't see too much of a performance differences at least when we test on DMS datasets. And this is also the case when we compare across models, right? So ESM2 actually does not give you, much higher performance compared to, for example, ANCA large or ANCA base or even smaller variants of ESM2, right? So I think it's sort of, again, this embedding, this latent space is sort of still a black box, right? So we don't have sort of good biophysical understanding about what it's actually doing. So it's hard to say, what's the next step of, for example, how to make this even better. And I think a lot of this will benefit by having more interpretable machine learning models.
Awesome. The next question I have, it kind of has two questions. So I'll read it, but we can definitely break it down. How well does the approach of training embedding work with low data setting? Since these embeddings are really high dimensional space, the number of parameters are still large. How do we prevent overfitting of these parameters since the number of data points are generally low? So basically asking kind of about like these low-end data regimes.
Uh-huh. Yeah, I mean, that's a super good question. And I think EVOLVEpro, if you think about it, at least when you deploy in the active learning fashion, right, you're forcing the model to overfit. So there's no way to circumvent the problem of overfitting because at least in the active learning format, right, in the beginning phase, you have very, very little data, right? You have 10 to 20 data points in the whole distribution, and you are just scratching the surface of the whole landscape. And I think this is why, these traditional statistical model, for example, random forest classifier and linear ridge regression actually work the best, right? So these model, when they are dealing with overfitting, they really start to shine compared to a lot of these, fancier machine learning models. For example, we also tried using a multi-layer perceptron MLP network, right? And regardless of how much parameter we trained, it actually always worked worse than these simple random forest classifier or ridge regression models. And I think, in the future, this idea of active learning and lab in a loop, right, where we start to explore this really vast design space, will always come back to this problem of overfitting because you never have enough data. The experimental throughput is not there, right? So the focus should be sort of focusing on the statistics and how models can deal with these regimes.
And just to follow up, they also asked if we can have an L1 regularized network.
Yes, I think there's plenty of room to sort of push the top layer regression model to be better. For our sake, we explored some very simple regression model, right? Ridge regression model, L1 or L2 penalized regression models, and we arrive at the conclusion that random forest classifier worked the best. In a very naive sort of comparison, right? And I do think having better L1, L2 penalization, and even better exploration versus exploitation strategies will certainly help.
Awesome. The next question I have, just because we're a little bit low on time, does the amino acid diversity matter for round one?
Sorry, can you repeat the question?
Yeah. Does amino acid diversity matter for the round one? So I guess the first pass of your active learning loop.
Yeah, so that's also a really good question. Yes, it matters. In the round one, what we find is that you want to maximize the randomness, right? You want to maximize the diversity of the sequence being explored by the model because you are sort of giving the model different seed sequences to start, right? And what we find is that, for example, in round one, if you were to use the informed selection, for example, if you use the mask marginal score from this language model, it actually leads you to be trapped in certain local maxima. So it's of course better to maximize diversity in round one.
And kind of going off of that, I think you just answered it a little bit more, but a follow-up question that was similar is how do you select the proteins for round one? I'm not sure if they mean you select what comes out of the model or if they mean like what you're putting into it. I'm assuming it might be what you put into it. So Ben, you can definitely unmute and clarify that, but it's probably like the starting point for round one, if I were to guess.
Yeah, exactly. So, comparing the strategies that we tested, right? It's always best to sort of just maximize diversity. So try different, mutations at different regions, and that's what gives the best performance.
Awesome. The next question I have says, for this study, you use DMS data for the training the top layer. Do you think the model can be confused if the training data set contains variants with multiple mutations?
Ah, so this is a very interesting and tricky question, right? We wanted to test this, right? So to ask the model whether it has the ability to sort of nominate and confer multi-mutant edits to a protein, right? And the problem with that is it's just really too hard to test and conclude whether the model has that capability because there simply doesn't exist a multi-mutant data set for you to benchmark, right? So all the public DMS data, at most they sort of have, combinatorially complete, 4 active sites for a given short protein. And, a multi-mutant distance for any protein is, of course, 21 raised to the n, right? And there's just no experimental throughput to measure the whole landscape for any protein, however short it is. So it really comes down to experimental validation, right? So in the wet lab, what we do is that after a few rounds of single mutation edit from a protein, we ask the model to rank, combinations of beneficial mutations for us to test in lab. And then only when we get the actual wet lab data, then we sort of evaluate how good it is. And at least in the naive settings, for what we tested, right? We just simply ask the model to combine the beneficial mutations and rank them. It seems to work reasonably well. But to say that the model have the ability to sort of directly nominate for you multi-mutants, I don't think it's quite there yet. And for a fact, we don't know. And it also speaks to the fact that we need more sequence to activity data.
Awesome. I'm going to close out with one more question. Just a reminder to everybody, you can join our Slack channel. And actually, Kaiyi has been very kind to say that he will join our Slack community and he can answer some questions asynchronous there. So if your question didn't get answered today, definitely reach out once he joins in the next week and get those. So for the final question for today, someone asked, how would your trajectories have changed if you didn't include the protein language model?
Ooh, that's an interesting part. So in the very beginning, right, I showed you this comparison between, protein language model versus, simple embedding. For example, one hot encoding, right, integer encoding. So these are, just regular statistical encoding of the protein. And you seem to always get a boost in performance, right? So meaning that the embedding generated by the PLM, they actually captured some high-level information of a protein, at least what they learned during evolution. That is not captured by these very naive embedding, for example, one hot encoding. So in that particular perspective, right, we will benefit by better and better large pre-trained protein language model, right? And we are now actually trying ESM3 to see how that particular embedding space compares to ESM2. And there's of course this speculation that we are saturating. So even if you make the model bigger, because the data is still uniref, right, and that's sort of stationary now, you won't get better or more information-rich latent space. But I think that's of immense interest to future work.
Awesome. Well, thank you so much for joining us today and talking more about your recent work. We really appreciate it. And for everyone who's still online, we will be back in 2 weeks with our next speaker. So thank you guys so much and see you next time.
Thank you.